I'm Phoebe Zarnetske. I'm here on main campus. Our lab is based here on main campus, but we do basically all of our field work at KBS and also some at UMBS. So our space here is actually just office space, and we rely on the excellent facilities at KBS to do all of our sample processing and things like that. Oh, okay, I just want to say, I forgot to put the little note here, but this is a picture that Kara Dobson took, and it won 3rd place in the LTER wide photo contest. I just took this as a screen capture, so it's not actually the high resolution version, but really nice photo of Rex early one morning this last summer. So congrats, Kara. You'll see this pop up a couple other times.
I also just want to say that the pictures that finished ahead of Kara were all from the Arctic sites, and I think there was some bias involved. So on my part, this was the first place winner.
Yeah, and actually some of the other ones were from my husband's the other Zarnetske lab that won first, one of the first or second place. So that was cool. So MSU was well represented. Good photographers here. All right. We're gonna talk about the work that we do at KBS, and that's primarily led by, we have 2 excellent technicians, Mark Hammond and Emily Parker, and grad students, Kara Dobson and Mariah Young, both of whom are PhD candidates. And then there's me, and I sit up here, but I work very closely, especially with Mark, to make sure that things run smoothly. So without Mark, I think this whole field research would really just not exist. And so I just want to give a shout out to Mark, especially for that. And we were recently able to create a position for Emily to stay on full time. And so we're really excited that Emily can continue on. She started as a URA and then transitioned to a technician this summer, and now is going to be full time with us. So we're really excited about that. I also want to mention that I'm the director and the lead PI for IBEAM, which is the Institute for Biodiversity, Ecology, Evolution, and Macrosystems. And that's a strategic partnership grant that MSU awarded to us. And the focus of IBEAM is really to bring together these different subdisciplines and to foster innovative research and synthetic research across those areas. So that's another hat that I wear here as well. And here's a nice picture of our one of our Rex treatments with a T-Rex decorated by my kids.
Most of my job is keeping the T-Rexes out of the experiment.
Yeah, it's a big problem.
No wonder Phoebe thinks it's a valuable work.
So and this is gonna be pretty informal. I'm gonna start by kind of giving you an overview of my lab and collectively what we do. And then we're gonna transition to what we do at KBS, but I thought it would be helpful for other people to know what else we do up here as well. So we're really, I think our motivating, our main motivation for research is trying to understand how biodiversity is going to be responding to climate change in particular. And climate change is just one of these leading causes of biodiversity loss. So there's, of course, these other main leading causes of biodiversity loss. And it's estimated now that there's about 1 million species at risk of extinction, and that's out of 8 million known species. Some scientists estimate there might be as many as 11 or 13 million out there. We just haven't cataloged them yet or found them. And so this is kind of like our big kind of motivation for our work. And you've probably seen some figures like this, where we look at the past global mean temperature, and we're about here, and we're thinking about what futures we are going to be choosing as humanity. And as an ecologist, I'm primarily interested in how the species, biodiversity, and ecosystem functions respond to this anthropogenic climate change. So, regardless of the future direction, there's going to be different responses by different players in these ecological systems. And so we really focus on not just individual species, but in particular how they interact with each other, how that influences ecosystem function and patterns of biodiversity as a whole. So my lab kind of has these three main focal projects ongoing now, which I'll just talk briefly about each: climate intervention biology, macrosystems biology and patterns of biodiversity, and then biotic interactions and climate change. And we address those main topics across different spatial and temporal scales. So this is a STOML diagram, and it's showing spatial versus temporal scale, as shown here. And I've just put some pictures on here of example projects we have worked on or are working on in the lab, going from, sort of more local and short-term studies up to more regional and global over longer periods of time. And we tend to focus our more short-term and local studies on trying to identify the underlying processes, what's actually driving potential responses of biodiversity to climate change, for example. And that's where these experiments at KBS come into play. But at the same time, we zoom way out and we look at global patterns, or we look at regional patterns or continental patterns. And oftentimes those are aggregated from data across broader and longer temporal scales. And so there's this constant kind of back and forth between looking at the pattern and trying to infer the underlying process, or designing an experiment that's trying to identify and quantify the process level influences, and then seeing what patterns emerge. So even though these can be in different systems, we use guiding community ecology principles to try and understand generally how these systems are responding to global change. And to do that, we combine a couple of different approaches: experiments, modeling, and field data. And it's really this sort of central area where they come together, where we have a lot of synthesis work. And so here's, again, just some of those examples of some of the data and some of the sources of those different approaches. And in terms of experimental work, so we conduct these climate change experiments. Oftentimes we have this kind of underlying kind of principle that warmer temperatures increase metabolic demands in ectotherms like insects. That might be you might expect that to increase their consumption rates. How does that then kind of exacerbate or play out at the more broader community scale in terms of plant influences, in terms of microbial influences? And so we do that in REX here at the LTER, but also up at the University of Michigan's Biological Station in a terrestrial system. But I've also worked with students, in particular Laura Tortocleb, who have worked a lot on aquatic systems and looking at how temperature mediates the predator-prey interactions between these aquatic insects. And another kind of driving kind of central theme that we focus on is this biotic multiplier concept. So a biotic multiplier of climate change would be a species that responds pretty strongly to direct effects of climate change, so like warming or changes in precipitation, and through their interactions with other species, then sort of imparts those changes even more, multiplies those changes across the ecological community. And so we're thinking a lot about which are those players in the community. Can we identify certain functional groups that play that role? Are there certain individual species that play that role? And so we design our experiments kind of with that theme in mind as well. So at the other end of the spectrum, we also leverage big data and ecology. So we're relying on, at these kind of broader regional to global scales, data that are not collected by us at all, but that are collected by many, many people, some of them citizen or community scientists, but also systematic surveys that are done, for example, with NEON, through LTER, the forest inventory analysis data. And collectively, those data represent just a huge amount of information, which, if analyzed appropriately, can provide some pretty important insights into really just quantifying the drivers of these patterns that we see, but also making us think about what are the potential processes that are underlying those systems. And a common kind of theme that we focus on at these broader scales is looking at the latitudinal diversity gradient, or just biodiversity gradients in general, whether they be in elevation or across latitude. And one of the main hypotheses that's sort of driving this distribution, at least at a global scale, is temperature. But because we're community ecologists and we understand that there's lots of other things at play, we know that it can't just be climate. And so we've actually developed some techniques through the macrosystems research to try and quantify essentially the influences of different drivers and how they interact with each other to ultimately explain patterns of biodiversity. And this role of interspecific trait variation is a pretty important one. So we think that the traits that are expressed by different species at in different communities and different locations within their range can help explain essentially the breadth of the niche breadth that they occupy. And that can be more directly related to a lot of these other drivers that ultimately can explain the pattern of biodiversity that you're seeing. So there's some kind of, in a way, like mechanism here that's represented by the trait variation. And so we're starting to do a lot of work with these trait values and connecting them then with these different drivers. Rather than thinking of climate as just directly affecting biodiversity, it has a lot of these interactions that we're trying to quantify. And then importantly, these are acting at different spatial and temporal scales. So there's a scale kind of layered in all of this. And we think that it's very important to acknowledge that there can be scale dependency in all of these relationships. And just quantifying this, for current day distributions of species is really important, because if we don't quantify it correctly and get these relationships correct, then any kind of forecasts in the future are going to be not quite right. And so we're truly trying to parameterize and perfect the models based on current day distributions so that we can make more informed predictions with climate change. And I should say most of this work involves some work with current and former postdocs in my lab.
Phoebe, I had a question about biotic multipliers, if you don't mind. I was wondering, are those generally keystone species, and that they're species that already have a large role to play in the ecosystem, and so a small change in their activity or abundance has a cascading effect? Or is it more species that are really, really sensitive to climate change, such that they're removed entirely from the system with a perturbation, or their traits change significantly? Like, are they usually species that were already kind of integral to ecosystem functioning, or is it kind of emergent with climate change where their effects come out?
Yeah, I mean, our kind of leading hypothesis with this is that it should probably map to species that are more like keystone species, but also in particular top predators, or anybody who's sort of a top consumer in the system. For a number of reasons. So top consumers tend to be lower in population abundance, so there are just fewer of them. Oftentimes they're just more sensitive, especially if they're an ectotherm or even an endotherm, sort of at the limit of their range. And because of their trophic level influence, then that can just by definition have a bigger effect down the food web. But on the other hand, you could also argue that some plant functional types would be especially important, because of course they're like the closest you can think of in terms of trophic levels, in terms of the direct response of temperature and precipitation on fundamental, photosynthesis and everything. So their response is quite rapid and direct. So, like thinking about the Arctic is greening up much earlier, and it's the herbivores who aren't able to keep pace with their migrations, like the caribou, with that peak foraging time. So in a way, it can also be these plant communities, these basal food web communities. So it's a concept to try and think about identifying species that might be most sensitive, but also have the biggest impacts. And I think those different categories of species could all fit in. And so we're trying to quantify the influence kind of with these different community modules that we're working with.
Awesome, thank you.
Mm-hmm. Yeah, good question. Okay, so one of the aspects in this figure is called geodiversity. You might be wondering, what is that? So geodiversity is basically a term to describe the inherent variation in abiotic features and processes on Earth's surface, in particular Earth's critical zone. And so that critical zone is what supports life on Earth, essentially. And you can also think of it as the stage upon which life exists. So life, can exist, but only in certain conditions. And it's that critical zone that really makes that possible. So if we go ahead and quantify the variation in geodiversity of various things like topography, even temperature and precipitation regimes, the idea is that areas that have high biodiversity should actually also have high biodiversity within their kind of level of biodiversity in that region. And so we've done a lot of work with satellite remote sensing to quantify geodiversity and then relate it to biodiversity. And Beth and Annie and Hazel have all worked on this in different capacities. And so we're basically showing that, yes, in many cases, you can find areas that are high in geodiversity, promote high biodiversity. So the question then becomes that, can we identify areas that have high geodiversity that actually might help buffer against changes in climate? So if they're inherently geodiverse in topography, in certain types of land cover, in soil type, et cetera, are those areas, because they're inherently supporting more biodiversity, can they be targeted for, let's say, reserve design or something, and might help buffer against, changes in other variables like temperature and precipitation? So this work involves a lot of work with remote sensing, and along the way we realized that we didn't really have a tool to quantify geodiversity per se. So we generated this R package, which basically allows you to quantify spatial heterogeneity in any gridded data. So that's shown here just for elevation, for example. And scale dependency is important because this again gets at these filters that might be existing on the landscape. So if we have a plot here, like a forest plot, or let's say we observe certain species, a set of like mammals in this area, we would want to try and understand if there's scale dependence in geodiversity. So one expectation is that the heterogeneity inherent in the landscape should be pretty influential at a more local scale than, let's say, a really broad spatial scale, like across all the state of Oregon. And that's sort of in relation to climate variability. Climate tends to have this sort of broader, footprint on being a filter to observing any species at a given place. But the inherent variation in the landscape features is likely to be kind of this more intermediate or more local scale. So again, we're doing the scale dependence analysis to try and understand this relationship. And we've so far found that, at least in the Western United States, tree diversity has this scale dependent relationship. Biodiversity has a scale dependent relationship with elevation diversity, so that you get higher diversity in trees at sort of this intermediate, local to intermediate scale. But after a certain sort of distance away from the plot, you no longer see that effect of geodiversity being very high. So again, we're trying to improve the way that we describe distributions of species, describe biodiversity patterns, and using these tools can help us get closer to that. So we also do a lot of work to fill in data gaps. Sometimes we start working on a project and then we realize, even though there's a lot of data out there, there's still some pretty big gaps. And so Laura really led this great project, really quantifying all of the different types of traits in aquatic organisms, aquatic macroinvertebrates in particular, across the lower 48. And this in and of itself is a huge data science component of our lab. So we work a lot to try and make reproducible workflows to pull data in from different sources, clean those data, standardize them, make sure that everything's taxonomically up to date, and ultimately then provide these data for the research community in open access publications. So this was a really nice contribution that Laura led. And then we also generate data in terms of, maybe we use remote sensing imagery, but we want to get some derived variables from that. So one of the things that's missing across the United States is a disturbance set of imagery. So at 30-meter resolution, we've been working on quantifying the disturbance frequency, severity, and then also the type of disturbance based on Landsat imagery. And that's been led by two postdocs that are former postdocs now, but finishing up this paper. And then just in terms of some of my own work that I've done more recently, I've started to work on network ecology. So in terms of statistical approaches, so while on sabbatical last year, I started this project with some collaborators at the University of Lyon. And we have basically connected different North American breeding birds, well, not just breeding birds, the breeding bird survey, but then I've been working on this database to basically delineate all the different types of interactions among these species. And the main question is to model how the networks of these species, so the connections between them, shift in space and time across the last 30 years and across all of North America. And our hypothesis is that they should be, if they're responding to climatic changes, we should see that emerging in the changes in the network itself, not just the distribution of the species, but the actual composition of the network, the roles that they play. And so we're kind of in the middle of that so far. And then finally, some work that I do with a postdoc is to work on this problem of climate intervention. So climate intervention is, broadly speaking, a term to describe means of either reflecting sunlight from the Earth's surface through injecting, for example, sulfate particles into the stratosphere, or having white roofs and white roads to basically increase albedo. It also can include carbon dioxide removal, so literally sucking carbon dioxide out of the air and burying it. Or, for example, planting trees. So that's another, sort of nature-based solution to that. So climate intervention can include all of those things. The working group that I co-lead consists of a bunch of climate scientists and ecologists, and we basically come together and talk about this problem and try to advance the research into potential climate impacts on ecology, of which there's just very little research right now at all. So this has been a really fascinating area to work in as well. So I wanted to just give that little overview of kind of the big picture, the different components we work on in my lab, just so you can recognize that we're doing other things that are related to the work that we do at KBS, but in some ways very different techniques and very different kind of motivations. But the questions, the overarching questions are very similar. How does biodiversity respond to climate change? What's the role of these species interactions? And I'm going to turn it over to Mark to go through some of the work that we're doing right at KBS.
Awesome. Thank you, Phoebe. Just before you turn it over to Mark, I just wanted to stop and see if anyone had any questions for that first overview part. It's a lot of cool stuff being presented. Mm-hmm. And also, I'm not the only one that's allowed to interject and ask questions.
Oh yeah, go ahead, anybody.
But yeah, Phil, go ahead.
Yeah, Phoebe, boy, lots of stuff to digest here. So can you comment on the geodiversity metric? And in particular, what's the smallest usable resolution for that? I mean, it appears that you've used it for pretty large areas, even regions or subregions within states. But could you use it on a on a smaller landscape level?
For sure. So because at their essence, they're measuring heterogeneity. You need, if you're using let's say a gridded data product, and the grid, the pixels in that could be 1 centimeter. They don't have to be 30 meters on the side per se. Because you're computing variability over space, you need at least, if there's a central location, you need 3 by 3 squares to actually make that measurement. So that would be the smallest unit would be 9 squares in total.
Okay,
But you
thank
could, it's basically gradient surface metrics applied to any surface, any gridded data product. So as long as you had, even on this plot, right, we could grid up the 1 meter by 1 meter square and measure, I don't know, soil temperature or something. And we could apply gradient surface metrics to that.
Sure. Okay.
So it's,
Great.
yeah,
Thanks.
very flexible.
Any other questions before we move on to Mark? All right, Mark, you can go ahead.
Thanks. So my name is Mark. I'm a technician. I work with Phoebe and Jen Lau. I've worked at KBS or on main site at MSU for over 20 years. I started my appointment on the LTR actually with Kay Gross, and then I worked with Doug Shemsky and then Jen Lau, who has some LTR work, and now with Phoebe. Most of my LTR work has been with the T7s, and particularly with the more micro plot experiments with nitrogen addition, and now with REX. I have a few slides to go over about our REX experiment and sort of more of a reminder of what we do. And there's a few towards the end about new thoughts that we've had this year and maybe are considering for next year. So I want to go through this relatively quickly to leave some time at the end. And if you aren't familiar with the REX setup, then please jump in and ask questions or save the questions to the end. For Phoebe's work, we early on, we met with Nick and we were able to add 2 additional footprints to the REX work. So the 5 common ones in the T7 are, I consider the LTR core footprints, and then we were able to add 2 from Phoebe's. And Phoebe's contribution, the biggest manipulative contribution is the warming treatment, which is the OTCs. So we have, in each of those footprints, we have 2 OTCs, and then we also add an insecticide treatment, which we hope to reduce the insect herbivores as a way of perturbing the plant community, essentially. And we also use the subplot control for the irrigated control footprint for sort of a reference for comparison against our other treatments. So that's in a nutshell, that's sort of Phoebe's footprints, and there are other researchers in other labs that are working with these footprints as well, which is really exciting. Grant's doing some greenhouse gas work, and Stephanie was doing some insect work, and the data are, at least some of the data are the same scale as the LTR core footprint, so it should be interchangeable for different questions. Next slide, please. So this is my favorite time of year. It makes percent cover really easy. This is after the spring burn, and this is a good picture of what a footprint would look like. So we have 2 OTCs, and the scale that we do most of the community plant research is a 1 meter by 1 meter survey, and those are the 4 quads. The OTCs in general, they passively warm the air temperature by trapping the radiation for a little bit longer. And depending on cloudiness, wind, sunlight, they generally raise the, on average, daytime temps 1 to 3 degrees Celsius. Next slide. So this is, before I started with Phoebe's lab, there were other researchers. Kylie was a grad student, and other technicians that, as well as a postdoc, that designed the OTCs. And this is a blueprint that led to a methods paper that details the warming and different conditions that the manipulation allows for. This basic design is the basic design is, you would find it in the Arctic. It's just smaller. So while OTCs have their the challenge is that it is a pretty well used manipulation across global warming biology. And we added a door, which is nice.
Actually, hey, Mark, I'm just going to say one quick thing, which is, so from that methods paper, oops, I'm sorry, I don't know why I'm not going back. Okay. We've had several researchers across the world reach out and ask us about this. So they've actually built them out. And as Mark said, we've modified and added a door. He'll talk about that in a minute. But our hope was basically to create something that could really, it's like literally a blueprint that people can take and then put together. And the construction material is pretty inexpensive. So it's, you know, really great for ecologists.
And they're also nice, particularly if they were a little smaller. It would take some work, but you don't need electricity and you don't need a lot of tools. So you could basically pack them into a field site. That's a little bit off the beaten path. So we have different, when we started this, we were aware from talking with Nick and Kevin about what their measurements were for their soil and air temp. So we've added some weather measurements that are to cover the OTCs. Most of them are hobo products from onset. So there's these pendants that measure air temp. We are using them at 1 meter tall. For 1 measurement per hour, there's 24 loggers spaced out across the reps. And we're also having a few humidity measurements, although I had some critters that were eating the cords. So field biology and science, interacting with chipmunks. Yeah, so let me get through this section and then we can come back to questions, Namir. Next slide. So what are we measuring? And Phoebe's interests are, I consider them that there's sort of a primary set of data collection, and then there's a secondary set, at least from what we started 2 years ago. And this was built up from her WarmX experiment, which is similar. It doesn't have a drought component, but it's an older experiment. It's 7 years old now. That's what she was describing for the University of Michigan's biology station. There's a site there. And then there's a site just by the soil pit. So our choice of measurements for REX came from that background, at least initially. So we measure phonology at a 1 meter square area. That includes green up in the spring, date of first flower, fruit maturation. We do some community assessments with visual percent cover. And also we do productivity, which on a smaller scale, and we do this with GENS, LTER core footprints too. And some of the productivity measurements are sorted to species, which then gets us back to some more community-driven metrics like species diversity and species richness that are a little bit higher resolution than just normal visual percent cover. Next slide. Phonology is, we can see it in the field in the spring, depending on the species. So it's exciting to me. I like to have be able to have visual responses. And the other good thing about it is we survey phonology about every 3 days. And it's been a really good metric to be inclusive with new field assistants, so undergraduates. We know what plants are on the LTR. We've done this before. So it allows new people into the lab to start to learn the plants, get out on the LTR, see these responses and collect data. So that's been it's just a pretty successful metric working with 2 seasons of undergrads to this point. And we do this over a 1 meter square area. And we're basically just documenting the date whenever any given species is flowering. And this captures the date of first flower and the date of last flower. And then we also document when we see fruit maturation start and then when we see it ending. Next slide. So then there's this sort of tier of what I think of as secondary measurements, at least they were when we started 2 years ago. We do some herbivory assessments, which we'll talk more about in a minute. We also collect some tissue for leaf carbon and nitrogen analysis. And we've wanted to do some measurements that are more physiologically important, particularly with the drought. We struggled a little bit with that. SLA is probably the one response variable that we picked to put more time into it, which may be indicative of a drought response. So these are, we do dedicate time to these, but we sort of, they're not as high a priority, at least to this point, compared to the other primary measurements and maintaining the treatments as well as supporting other work. Next slide. So I guess real quick, that was sort of an overview of what we've done before. I think most of you have seen that. It was similar slides from the Rex meeting in March. And Phoebe's lab has talked about the herbivory work, and we're hoping to dedicate more time to that. And one of the thoughts is it's a top-down driver for the plant community, particularly for if we're getting herbivory on the dominant species. So in some ways, this is sort of it has an experimental design that is top down versus bottom up, if we're thinking of water and nutrients from the soil as the bottom up factors. And this is sort of how I describe it to undergrads that are coming into the lab. They've probably heard about these as trophic level responses, and it has some community level data. And so herbivory is, we don't see a lot of herbivory on the leaves with the Warmex, and that's, we see enough of it to work with it. But we see a lot more in Rex experiment with clover, milkweed. There's a couple of aster species that have lots of herbivory, and so that's more promising, at least to me. We see a lot more of it, so the resolution of that data we're hoping is a little bit stronger. That enables us to ask a little bit more in-depth questions in the future with herbivory. So that's sort of a change of possible priority leading into next summer. Next slide. And that led us to how we measure herbivory. We've talked with Will Wetzel's lab, particularly with Luke, and they have so those of you that aren't familiar with Will's lab, he's on main campus. He's an entomologist, among other things. And he's developed this, he calls it HerbVar. There's a website. It's a national network. We're looking at herbivory on plants. And this was our first summer that we made some measurements comparing our old system of measuring with Will's protocol, which is more involved and involves counting more leaves and getting plant size measurements. So at the end of the day, Will's allows us to have a severity of herbivory and also a more general measure of herbivory on the plant that's determined by its size. So we're hoping to introduce this data more thoroughly next summer, at least on a few species. And that may also mean that we're spraying insecticide more frequently and being a little bit more aggressive with that treatment. To this point, the last 2 summers, we have made insecticide treatments, but it's sort of taken a back seat to other research needs. Next slide. So this was literally data that came in a week ago. Emily and I went out and we're counting herbivory on goldenrod, so Solidago canadensis or Solidago altissima. And we do see this damage a little bit on the Warmex, but we see it a lot more on Rex, probably from grasshoppers. There might be some other herbivores, and most of the plants that we surveyed do have herbivory, and the severity of it is present and variable. So this graph shows essentially most of the plants are having at least some level of herbivory, and some plants have almost every leaf eaten, at least to a small degree. Next slide. And this is the severity or leaf area damage that was measured on average. So on average, the highest level is around 12%, and about 40% of the plants that were surveyed have that level of damage. And that's exciting to me because with the Warmex work, which we're writing a manuscript up on now with Kara and Mariah leading that, the herbivory levels on Warmex for Solidago were much smaller than that. And we are seeing interesting results for that study, but with Rex, there's a lot more herbivory. So that might be an interesting ecological trophic level to look at more in the future. Next slide. Lastly, for me, one of the parts that I really enjoy a lot with being a technician is working with undergrads, and the LTER research, it's been a really good fit for undergrads and for Phoebe's lab and for me as a technician. We've had 2 summers and we've had several undergrads. Kristen was a URA. Emerson is a K College student that started off as a high school volunteer and working with Jen, and she's stayed on the lab part-time for 3 years now. Lee was a RU with Mariah this summer. And then Emily can introduce herself more in a minute, but she started off as a URA and it's done really well, presented at URAF. And then Jordan was this summer's URA, and he's stayed in the lab and he's excited to present his first poster at URAF for this first more larger scale poster presentation. So that's worked really well. Last slide. So this is to Emily. So I'm kind of making a transition to a different speaker. Let me see how we're doing on time. Any general questions about Rex and Phoebe's lab's work with Rex?
YJ, did you want to unmute and ask your question?
Yeah. So Mark, I just want to know if the OTC affect the herbivory. I mean, because it looks like a barrier between inside and outside, right?
Sorry, whether it affects what?
Affect the herbivory.
Oh,
Yeah.
we haven't analyzed the data yet. We have the goldenrod data. There's a data set that we'll be able to test that question. We do see grasshoppers in it's an open top, as you know, and there's a little bit of space on the bottom. So the space inside is accessible to herbivores, particularly those that are able to move quickly. But that is a concern of whether it's an artifact and whether it's influencing either the numbers, or it also may be a safe haven for them against some of them against other predators. So it's sort of an open-ended question right now, but we are getting some data to test that question.
Yeah, I could just jump in on the Warmex experiment. We don't see any evidence of that, although we don't necessarily we don't look at the insect community per se, we're looking at the result of it. So we're measuring herbivory. So it's possible that the players may shift depending on the treatment. It's just we don't have the data, like insect community level data to look at that.
Awesome. Thanks for that question, YJ. Thanks for tuning in from all the way across the world too. Awesome. If there are no more questions, then we can move on to Emily. And yeah, Nick says, thank you, Mark, for working with all the undergrads in all of our labs, which I will second as well.
You're welcome, Nick.
All right. Hi, everyone. I'm Emily. As was mentioned before, I was a URA with the lab in 2021, and I'm now a technician with the lab. And my undergraduate project was investigating how the climate treatments that we use in REX affect the goldenrod gall maker system. Particularly, we looked at the goldenrod bunch gall midge, which makes the characteristic rosette galls on top of the plants. It's not a very well studied system compared to the ball gall that's found lower on the stem. But I was curious to see if the warming or drought treatments had any impact on both the plant and the insect gall maker. So we measured a lot of different things, both for the plant, like plant height and phonology, as well as for the insect mediated traits like the gall size and the larval chamber size. And this is just some preliminary data from last year. We collected more data this year to kind of bulk up our data set, and we are working on writing a manuscript on this soon, hopefully.
Awesome.
And then, okay.
Yeah, hi everyone, I'm Kara. I'm one of the PhD students in Phoebe's lab, and kind of the overall theme of my research is looking at how plants respond to experimental warming and drought. And the main questions that go along with this is looking at how do plants respond to climate warming at both local and global scales. So locally, I'm looking at that both with our WarmX experiment that Mark talked about at KBS that's kind of adjacent to the LTER, and also that experiment is replicated up at UMBS, which is that lower right photo. And like Mark talked about, we look at various plant responses to warming in that local scale, like phonology, etc. And then at global scales, I'm conducting a meta-analysis of warming experiments across the globe to see how plants respond to warming all over the world. And for that meta-analysis, I'm also looking at potential drivers of these plant responses to climate warming. So things like species-specific differences in how they respond, plant functional group differences, and also things like if latitude contributes to how these plants respond to warming, and also lots of other things like the amount warmed by the experiment and other factors. And then secondarily, a specific plant trait that I measure that I'm looking at in the LTER is how plant volatile organic compound emissions are affected by warming and drought. And these VOCs are chemical compounds that plants emit to use for things like plant communication and interactions with insects and other organisms. So I'm interested in seeing how these interactions between warming and drought affect the ability of plants to emit these important compounds. But overall, I'm really just interested in these various plant responses to warming and drought and what might contribute to those plant responses.
Okay, and I think Mariah is traveling. Yeah, so I'm just going to quickly say, kind of summarize Mariah's research, but then I'd like to open it up for questions, particularly for Emily and Kara who are here. So Mariah's research, her overall theme is the interactive effects of climate change and soil microbiome on below and above ground trophic interactions in these communities. And so her main questions are, how do bacterial and fungal communities respond to multiple climate stressors? And does the soil microbiome mediate plant growth and phenological responses to these stressors? So I think that's the last slide. So we can open it up to questions for the last little bit.
Thank you all. Yeah, if there's any questions, then feel free to post them in the chat or just raise your hand and unmute. I had one question for Emily, which was just, what's your what's the hypothesis behind how warming might affect gall formation? Like, is it that I guess, why would you, is it supposed to be a change in the plant that's making them more susceptible, or is it a change in potentially the insects that's making them want to form more or less galls that you think is at play, or that you all have discussed being at play?
Yeah, that's a great question. So galling is pretty interesting in where the size of the gall is mediated mostly by the insect rather than plant traits. So one hypothesis we have is warmed galls might be smaller because, as Phoebe mentioned before, that warmer temperatures kind of speed up ectotherm life cycles. So that's a possibility. But there's also another hypothesis would be that the drought and warmer temperatures could stress the plant out and leave less nutrients available to form bigger galls. So I think overall, we'd predict that they would be smaller. And I can't really speak to like how many galls would be formed because that's kind of a tricky question. But yeah.
Awesome, thank you. Any other questions from the group? We are running close to time, but we do have time for a question or two if there are any. Grant says the gas flux cylinders regularly trap spiders. Maybe there's an experimental design question or a possible hypothesis to test there. Phil, yeah, go ahead.
Yeah, another question for Emily. Emily, I see there's a pretty strong warming response to plant height. Is that a biomass response as well, or is that primarily height? And physiologically, what's going on there, do you think? Is it a earlier emergence or what?
That is a great question. Mark, do you have any insight on like ANPP and how that's affected by warming?
We do have those data. I don't recall them for Rex. And we're actually, this summer's data, we have the plants, but we haven't weighed them yet for ANPP. For WarmX, there was a trade-off, and that plants in the OTCs for goldenrod were much taller, but there were fewer, their density was much less. So there was a trade-off between the growth of the plants gaining more height, but producing fewer ramets. I would expect the plants to at least on an individual plant basis to have a higher mass for Rex in ones in the OTCs, but I don't have those data at this moment.
Yeah. Huh.
Other questions? We've got about a minute left before we got a break.
Could I have a question?
Yeah, go ahead, YJ.
Oh, it's a additional question from Phil's question. So I don't know when is the measurement, the time of the measurement. Is the warming just speed up the growth or also increase the whole growing season production?
So back to Phil's, I didn't fully answer that with so we have green up data on goldenrod as a species level. So their emergence, they do show up and grow. Early green up is earlier. We have measured height a few times during the season. We haven't measured them frequently enough to have a growth response curve. We're not measuring height every week or 2 weeks, but maybe 3 measurements through the full season. And my prediction would be that the warming, at least in the spring, is earlier, and then possibly their growth is faster in the spring, and then the ambient plots do catch up to their height later on. I think that the warming is certainly warming plus drought is a stress, but warming by itself, depending on rainfall and phonology and ontogeny of it could be either have a beneficial effect to the plants, or it could be detrimental depending on the rainfall. So does that answer your question, YJ? So the short answer to your question is we do have those data, but it may be suggestive to answering your question, but we did not design the experiment with data collection to generate a growth response curve.
Thank you. Thank you.
Thanks.
Nick, did you have something you wanted to say before we sign off?
Yeah, I do. And I was excited to see an email land in my inbox last night announcing the College of Natural Sciences award recipients, and so it's hot off the presses that Phoebe won the Early Career Research Award for the college. So it's a particularly fitting time to hear all the great work happening in her lab. Congratulations, Phoebe. I think she gets the award in like 3 hours from now.
Yeah, exactly.
That's awesome. Perfect way to end. But yeah, thank you, Phoebe, Mark, Emily, and Kara for presenting all this work. Really awesome to get to hear what y'all's lab is up to. And yeah, thank you everyone for tuning in. Really appreciate it. Great questions, great turnout, and stay safe out there in the snow.